Hey, Braves. So I've got a little one in the other room, so I'm going to try to be quiet. Hopefully you can hear me. I've got it connected to my earbuds here so I don't have to talk too loudly. Today, what you're going to be doing is a pumpkin lab. It's the anatomy of a pumpkin lab. And this lab might seem silly at first, but there's actually a point to it. It has to do with what you are learning about plant structure. If you keep in mind, you are learning about the different types of cells, sclerenchymal, collenchymal, parenchymal, those three different types of cells. Well, they make up most of the tissue um, of your different plants and different plant parts. And the pumpkin is the reproductive organ. Um, Essentially, it's the progeny of the plant. And at the bottom of the pumpkin is where the blossom used to be. And the pumpkin itself is actually a ovary. And inside of the ovary are the seeds, the fertilized egg cells that became the seeds. The ovary of the flower actually grows into the fruit and the flesh of the fruit. So when you are eating an apple or something like that, you're actually eating the ovary of the flower that then grew after fertilization occurred. So, anyways, you're going to be looking at the insides of these pumpkins, and you're going to be drawing what you see. You're going to be labeling all the parts, and then you're going to be required to explain the different types of cells that those parts are made of. Now, it might not just be one part. For instance, the seed itself has several different types of cell types. Now, how are you supposed to know this information? Well, the assignment, and I should have had this pulled up to agricultural ecology, sorry. The assignment that you've been working on for the last week has this information about those types of cells. So when you go to the plant structure notes assignment, um, I do ask you to list the different types of cells and all of that. But on here, you will find information regarding this exactly, the sclerenchymal, the collenchymal, and it's going to tell you what they're typically used for. And if you just take a look at the pumpkin shape and all of that stuff, hopefully you should be able to figure that out. Now, just in case this is too difficult, you're having a hard time, maybe you're having a migraine today, like I did this morning. Well, there's something else you can do. You can type in, once you know the parts of the pumpkin, you can type in what cells are the endocarp made of. And Google will be able to direct you to the correct website to figure that out. Now, you're probably asking yourself, well, how am I supposed to know the different parts of a pumpkin? Well, for that, what you want to do is go to Google. So when I go up here, I'll just type in straight to my search bar, anatomy of a pumpkin. It's the name of your lab, right? And when you type that in, you're going to find in Google image search something like this. You've got your skin, which is the epicarp. You have the pulp, which is a mesocarp. You have the seeds. You have the fibrous strands um, that are the endocarp. So think about what these things are, like, stringy, fibrous stuff. Well, that's actually the description of one of the types of cells, collenchymal, right? That makes up the stringy bits in celery. So you would know that, okay, well, the gross stuff, the fibrous strands, the guts, are typically made of collenchymal cells. Okay, well, then what about the mesocarp? Well, you can actually go and type in Google, what cells are the mesocarp made of? And you got to do a little bit of reading. You're looking for one of the three, right? So keep reading until you find that. I have to click on a few websites, but that's what research is about. That's being human, right? Okay. So, anyways, this is kind of the lab today. Nothing too crazy, pretty straightforward. Other things on this, I'm asking, like, what is the purpose that each of these parts serve? There is a purpose, obviously, to the seed. There's a reason why the insides of these have that fibrous strand and whatnot, those fibrous strands in there grabbing onto these pumpkin seeds. So if you don't know the purpose of these things, you can use Google. It's okay. It's okay to do research. That's part of being a human again. Okay. So anyways, Now for the types of pumpkins and then what to do. So there are 3 different types of pumpkins up here. This is important that you pay attention to this part because I don't want you to get this mixed up because you're going to be cleaning out these seeds like the other classes did, just like right here. And when you're done with cleaning these out, by the way, this is where your spent pumpkins are going to go. But I need you to know the different types of pumpkins so that you put them in the correct location. So notice these seeds have been completely cleaned. There's no pumpkin residue left on them. They've been washed and then they've been left out on this paper to dry. This is for the sugar pumpkin. They all have it labeled at the bottom of the paper, so it should be pretty straightforward, y'all. Um, the sugar pumpkins are your pie pumpkins. Those are the 2 smaller ones that have the nice thick stems. You have a white pumpkin, which is also known as a cream pumpkin. There's different names and versions. I'm not sure which one this is. It's just white pumpkin as far as we're concerned. And then you have a jack-o'-lantern, okay? So whoever gets the big jack-o'-lantern, that's what it is. Make sure it gets put over on the regular pumpkin section, all right? This is regular pumpkin, the jack-o'-lantern. These are the pie pumpkins, the sugar pumpkins, and then you've got the white pumpkin. Okay, don't get those mixed up and make sure that your pumpkin looks like these when you're done. You can go ahead and put the guts in here after you've cleaned them off the seeds. Wash the seeds out with the colanders, rinse them really good, really get in there and get all that junk off because otherwise the seeds will mold and they will be useless to us. Okay, so that is your lab for today. Now, what to do after that? Well, you know what the answer is. You have the plant notes, plant structure notes assignment that most of you haven't finished yet. So finish that up in the notebook. Today, what I was going to do if I was here, and unfortunately I'm not, trust me, I did not want to be gone today. There were too many things that I needed to do for you today as part of these lessons. But what we were going to do today was go over zinnias as a plant helper, and I was going to explain stuff all the way down to the type of cells. So we were going to talk about lignin and things like that. That's something that I'm going to have to get you caught up with, but thankfully you're my 4th hour class, you're my longest class. So anyways, really quick, here's a note for you. Parenchymal, collenchymal, and sclerenchymal cells. These are the 3 for your lab. Parenchymal are used for storage. They're typically the fleshy parts of fruits and things like that. You can find it in the stem, you can find it in the root, inside of leaves, and inside of the pulp of fruit. That's really where the parenchymals are located. Again, it's the storage mechanism. Collenchymal, those are stringy, fibrous bits. So anything that's stringy and fibrous are collenchymal. And then your sclerenchymal are typically dead cells, but they're also typically made of a lot of lignin, which is a really dense, tough material that the plant makes. So if you have a part of your pumpkin that's dense and tough and made of dead cells, maybe that clues you in, right? Okay. So that's it for today. If you get all of this done and you guys have been cool, then the sub can go ahead and play an episode of Ancient Apocalypse if you want to watch that. But otherwise, I will see you all tomorrow, hopefully. All right. Be good for the sub.